Sweet. Okay, cool. All right.
Perfect.
Thanks for having me. And yeah, so I'm going to be talking about using machine learning to identify co-geography between Anopheles and Plasmodium. Um, so I don't think I need to convince anybody that like vector-borne diseases are a big deal, and malaria in particular is a pretty big public health threat. And biology-wise, it's also a pretty neat system because we have like at least 3 players here that are all kind of interacting with each other. We know a fair bit about the interactions between humans and Plasmodium. Like sickle cell is kind of a classic example for a reason. And we also think a lot about how humans and mosquitoes interact in terms of things like pesticides, preventative measures, gene drives. Um, but when it comes to the dynamics between Plasmodium and Anopheles, it's still not quite clear what's going on. We don't have as clear of a picture. And it's important to understand kind of the whole interaction, all 3 guys, to get the whole picture. So with that in mind, today I'm going to talk about what we can learn from the system using supervised machine learning to predict location from genetic data. In particular, I'm going to be talking about using inference errors to identify common spatial structure between mosquitoes and malaria. I'm going to talk about the different ways that we can interpret those errors and how we can be sure that they're because of biology. And then finally talk about, what could be causing this and what to look at next. So very quickly, I'm going to talk about how supervised machine learning works. So with this method, we start with a labeled data set. So here, for example, I have pictures of lizards and pictures of mosquitoes. This is the training set, and this is getting passed to a model that is going to learn the association between images and labels. So when this model's trained, we can give it a new unlabeled piece of data, and it will predict a label for that new thing based on what it's seen in the past. And this method is really useful for biology. So in this example, our data were images, but you can imagine replacing those with something like genotypes, or perhaps even, using genotypes that represent certain evolutionary scenarios. So this is a really powerful method, and there are like a ton of examples and papers about different ways that you can apply this. Today, I'm going to talk about a method called locator, that predicts a geographic location from a genotype input. So how this works is we start with a set of training data that consists of individual genotypes and then their X and Y locations on a map. So that's our training set, and our network is going to learn to associate genotype with location. The network sees a genotype, does a bunch of linear algebra, and then predicts X and Y coordinates for that genotype. And during training, the goal is to minimize the distance between a true and predicted location for all the data. So that is what we call the loss during training. And then finally, once the network is trained, we can give it a new genotype and it'll pop out a location that is representative of where it thinks it'll be. And when we apply this to simulated data, it works pretty well. So here I'm showing you just true locations connected to their predicted locations as per generation dispersal rate increases. So obviously we don't see as good performance when there's less spatial population structure, but overall pretty good. And we also get pretty good results with relatively few training samples and SNPs. So when we apply this to empirical data, we also see pretty accurate prediction, which is exciting. So here I'm showing you the HGGP data set. And overall, predictions represent where those individuals are from, but there are some outliers with really big errors. So we were wondering, like, what's causing that? Zooming in on the individual with the biggest prediction errors in this data set, this is somebody with Chibo ancestry. Their errors kind of project towards Eastern China, which is where that ethnic group lived prior to the 18th century. So those errors are kind of pointing towards ancestral locations. If we go back to our mosquito friends, Anopheles gambiae, we again see some outliers in error, particularly in West Africa, which is also where there's a lot more mixed population structure. So we think that when there are errors in location prediction, they're telling us something about how genomes are moving through space and time. That data set has gotten bigger since that paper, and we still see these really big prediction errors, particularly in West Africa. So here I'm showing you true sampling locations in green, connected to mean predicted location in blue. And when we apply locator to Plasmodium falciparum, the malaria parasite, we see similar error patterns, these really big errors in West Africa. And in fact, if we pair spatially proximate samples across the two data sets, we see that the error vectors are correlated, which is kind of exciting. So what's happening here? Why are we getting similar error patterns between the two species? And so what we think may be happening is that we know mosquitoes are going to move across the landscape, maybe not that far, but they move. And since Plasmodium is an obligate parasite, if one is infecting a mosquito when it moves, it's going to move with them. And so when these guys end up in a new location, they're still going to be more genetically similar to the populations that they originated from. And so we're going to have similar error patterns. So that's a cool biology explanation. However, bias in machine learning is a really big problem. And if you notice, the majority of our training data also comes from this region. So are we getting errors because of biology or just because our network doesn't train very well and is being biased? So to investigate this, I used SLIM. I simulated first a very rough approximation of Anopheles randomly dispersing across their range. And the key here is that they're randomly dispersing. So the degree of population structure should be smooth across the landscape. I sampled these guys with the same distribution as the real data set and applied the same locator method. So here showing you those empirical predictions again, and then the simulated ones. So we can see that the predictions are much more accurate in our simulated data set, particularly in West Africa, where we see those really big errors that we were worried about. And we can see also like very different distributions in terms of the magnitudes of prediction errors. So we feel pretty confident about what's going on with these predictions, but this is a method that we want to be able to be applicable out of the box for all sorts of species, and so we needed to kind of test the limits of prediction bias. So again, I used SLIM, this time again simulating randomly dispersing populations, again so that variation is smooth across the landscape. All errors should be about equal. From these simulations, I made artificially biased training sets, so sampled more densely from the right half of the landscape. And then after training locator with these data sets, use them to predict on a uniformly sampled training data set, not training set, sorry. And so doing this, I asked whether or not predictions were going to be biased towards the right half of the landscape where sampling was denser. To do this, I kind of dissected prediction errors into both x-axis and y-axis prediction errors. The idea being that if predictions tend to be biased towards the right side of the landscape, then this x-axis error will tend to be greater than 0. And indeed, that is what you observe, unsurprisingly. So as training set bias increases along the x-axis here, we can see that the x-axis error does get higher above 0. And this effect is a lot more pronounced with higher dispersal rates. Obviously, this is what would happen, but is also not a super ideal performance, especially because when we have real data, it is not evenly sampled across the landscape. So we set out to account for this bias. And to do this, recall that during training, the network is aiming to minimize this loss, the distance between true and predicted location. So with our bias training sets, we assigned a weight to each sample that was inversely proportional to the density of sampling around it. So a sample from the left half of the landscape is going to be important, a sample from the right half of the landscape, less important. And then during training, these sample weights are multiplied into the loss, so basically making the network pay more attention to sparsely sampled regions during training. And we see that, while it does increase the variance in our predictions, we do eliminate, for the most part, the bias. So that was exciting. There's still some bias in extreme cases, but overall, we see that we can reduce that x-axis error. And adding in the downsampling in pink, which is kind of a naive method to deal with spatially biased data, we see that our approach works much better, and it actually counts to keep all of the data. So to revisit our host-parasite situation, I've added the distributions of error magnitudes beneath the maps here. We applied sample weights to these data sets. and saw reduced overall error. So the original distributions are now shown in dotted lines, and I'm showing you the weighted predictions. And we still see those really big prediction errors, particularly in West Africa. And they're also, again, correlated. So we're feeling pretty confident that we're seeing something that's due to real biology, not just bias in the data that we have. And what we think this correlation is representing is this paired movement across the landscape, where you end up with samples that are both more genetically similar to another location. And we can identify this pattern not just with supervised machine learning, but actually by paying close attention to when it makes mistakes, which is pretty exciting. So there's a lot to be uncovered in genomes with these sorts of methods that maybe we wouldn't be able to see on our own. So just to recap, there are multiple reasons for prediction errors when we're using these sorts of methods. Here we have both spatial bias and population structure. We developed a method that reduces the impact of that bias on these predictions, and use that to identify common spatial structure between Plasmodium and Anopheles. So the next things that we're interested in looking at with the system is kind of like what genotypes are moving across the landscape. Are particular ones moving together more often? Are they epidemiologically relevant? We're also interested in how human movement contributes to these paired migration patterns. Obviously, we move farther distances than mosquitoes do, and human migration corridors tend to align along those same axes of error that we see. And lastly, looking to see if we can identify these paired movement patterns in other parts of the world where there are Anopheles and Plasmodium. So with that, I just like to thank my lab, Andy and Peter, my co-authors on this. CJ made the original network, excuse me. And Nate and Scott helped a lot with chatting and ideas. Yeah, so with that, thanks, and I can take any questions.